It is my honor and delight to introduce our speaker, Melody Swartz, today, who's going to talk to us about immunoregulatory roles of lymphatic vessels in cancer and opportunities for immunoengineering. But before I bring her to the stage, I want to just go through why we are all gathered here. So I'm the director for the Institute for Engineering and Medicine, and every year we have an Otto Schmitt Distinguished Lecture, and you can see some of the background on these slides of Otto's legacy. He was a faculty here in electrical engineering. He's the creator and co-inventor of multiple technologies, including the Schmitt trigger, but he's worked in biomimetics and biophysics and bioengineering, and he made significant contributions to a wide range of fields. He even worked in submarine detection, electrocardiography, and quality of life measures. You can see some of these here outlined on the slides. So the reason why we're having the lecture series is really to foster discussion of the grand challenges for engineering in medicine, and we invite national opinion leaders, of which Melody is one, in research and industry and government to give this talk every year. So before I call Melody up, I need to say a few words, and I'm trying to join the 21st century here, but not quite making it, so I'm going to do part of this by actual note. So Melody is the William B. Ogden Professor in the Pritzker School of Molecular Engineering at the University of Chicago, where she holds a joint appointment in the Ben May Department of Cancer Research. She obtained her BS from Johns Hopkins and PhD from MIT, both in chemical engineering, and carried out postdoctoral work at Harvard Medical School and Brigham and Women's Hospital. She started her independent career as an assistant professor at Northwestern University in the Department of Biomedical Engineering before moving to École Polytechnique Fédérale de Lausanne, EPFL, where she was promoted to full professor and eventually served as the director of the Institute of Bioengineering. Trained as a bioengineer, Professor Swartz uses quantitative approaches in immunobiology and physiology, including biotransport and biomechanics, to develop a deeper understanding of how the lymphatic system regulates immunity in homeostasis and disease, particularly in cancer and chronic inflammation. Her lab applies this knowledge to develop novel immunotherapeutic approaches in cancer, including lymph node-targeted vaccine approaches, as well as in vitro model systems that recapitulate relevant features of the tumor immune interface. Among her many honors, Swartz was elected to the National Academy of Medicine in 2020, elected to the American Academy of Arts and Sciences in 2018, and was named a MacArthur Fellow in 2012. Please join me in welcoming Melody to the stage.
Thank you so much. I guess I'll take this off. I'm very honored and humbled to have this honor of giving the Otto Schmitt lecture, and I'm very grateful for the invitation. I also am grateful that you used a picture of me that's more than 10 years old. It's not, yeah. Yeah, when I read about Otto Schmitt and what this lecture is about, really talking about what are the grand challenges for engineers in medicine, I thought I would say a few words before I talked about lymphatics on the overall concept of immunoengineering and why I think it's really the time right now in bioengineering for it's the time in immunology has become ripe where engineers can really start to come in and make a big difference now. And so, of course, immunoengineering is just engineers working on immunological problems. But we can think about, engineering tools, engineering systems like drug delivery systems or vaccine delivery systems. And it comes from a lot of different types of backgrounds, but I think the other half of immunoengineering is really doing basic immunology just from an engineering perspective. the goals of this is obviously to enable or facilitate clinical translation, but also to bring fundamental new insights. And so translational immunology has grown amazingly over the last several decades. Immunology has been around for a long time, but we've gotten to the point where things can start being translated now. And if we look at just in the last 20 years, so many diseases have been discovered to be autoimmune diseases. New antigen targets have been identified. Allergy and asthma is just rising incidence, right? And cancer, there's a whole new revolution in immunotherapy. And so really the basic immunology that has led to this is now ripe for translation, I think, and this is where we really need engineers. Immune modulating drugs are among the fastest growing pharmaceutical sectors. Not just, we think about all these diseases. A lot of, for example, autoimmune disease, we treat with steroids usually. We're not curing it, we're treating the symptoms. Same with allergy and asthma, we're not curing them. Cancer, we hope, these new immunotherapies have the hope to actually cure these diseases. Of course, translational immunology has been around forever, well, at least a very long time, and vaccines are arguably the most successful medical intervention among maybe antibiotics, maybe one would argue. And, since the 1700s, late 1700s, we've been vaccinating. And so it's a crowning achievement, really. And it used to be where, people would want to get vaccinated because people could remember or experienced very bad outbreaks, such as the polio, which devastated children in the 1940s. But most of these diseases, as they come up, we have developed vaccines against them. But even with vaccines, there are still a lot that we don't know. So we have good vaccines against many viruses, but there are still things that we've been working on for decades, and we still can't develop a good vaccine. HIV is a good example. So why haven't we? Why, 3 decades and billions of dollars worth of effort in HIV, why haven't we? Turns out, well, immune regulation, as you know, is really highly complex. It's not just a matter of self and non-self. We have to recognize non-self that's safe, like food, pollen, cat dander. We have to also recognize self that is disease, like cancer. And so it's these, when pathogens can take advantage of some of the physiological mechanisms that we use to differentiate these things, those are the ones that are the most difficult to vaccinate against. And of course, immune regulation, because of this, is highly complex. And in the last 20 years, like I said, there's just been so much more that we've understood. We now know that, for example, different immune cells play diverse roles and interact in complex ways depending on the situation, and more immune cell subsets are being identified almost every year. We know a lot more now about molecular signals, such as what, where, and how, that give instructions to different immune cells. And different pathogens require different kinds of immune responses, and they can hijack these immune cells in different ways. And then most close to my own heart is really the whole system of the immune system, which is the location and timing of cell interactions that shape acute response and chronic inflammation or chronic disease, and also helps shape immune memory. So dysregulation over long periods of time can lead to things like cancer and autoimmunity. But your immune system is constantly working to stay robust against foreign pathogens, but maintain tolerance to self-antigens. And this gets more and more difficult as you get older. That's why autoimmune diseases are often late onset in cancer. Okay, so we now understand these things, and so now we understand them enough where we can start developing translational approaches. And engineers are really bringing a lot to the table, not only in helping identify some of these mechanisms with various tools, but also by helping translate these things. Okay, so the hope overall is immunotherapy can cure chronic diseases, and so this is a really ripe time. It's an exciting time. And so immunoengineering really brings a bridge between the basic and the translational in the clinic. We can develop tools and approaches to monitor, predict, and control. All kinds of different work is going on, and this is what we really highlighted. When I moved to Chicago, it was to start a center and a big effort in immunoengineering. And we have established that now. It's it's the Chicago Immunoengineering Innovation Center, and we focus very much with working with clinicians and on human problems, human immunology. So an example I just want to give is in cancer immunotherapy from my own lab. Tumors can recognize, we can get, a tumor grows, you get an immune response, but there are so many different ways that tumors can escape the immune response. And these have been studied, as I said, in the last 20 years. We now know so much about all these mechanisms. And really, scores, I would almost say hundreds maybe, but I decided scores was maybe safer, but different targets are being explored clinically. And you can look at all these things I've listed, like antibodies. Sorry, it's so small on my screen, but things that would activate T cells or promote their activation or prevent their exhaustion, all the way to molecules that would help antigen presentation and education. There are, VEGF, there's CARs, there's all kinds of cytokines to help bring more immune cells into the tumor. And even the approved immunotherapies only benefit a very small percentage of these patients. And so for all of these new therapies, we have there's been checkpoint blockade for so many different cancers that we have enough clinical data that we know what correlates with response. But what if we want to start making combination drugs with TGF-beta inhibitors to target Tregs in the tumor environment? Or what if we want to bring IL-12 to help enhance DC in situ activation of T cells? Or what if we want CCL4 to bring in more dendritic cells? We don't know what kinds of tumors will respond or what kinds of patients, and there's so many things being developed that it's kind of mind-boggling. And so my lab has been working on predicting methods to predict which targets are best for a given patient. And we do this with a mouse model, and I know that this is different than a human, but the idea is that in a mouse, we have, in a syngeneic mouse, we have a tumor growing and a tumor-draining lymph node. We let it grow to full size. And then the idea is to take it out and make these in vitro avatars. And then we can do all kinds of screening, and then we can go back at the end and correlate what we find with the type of tumor. And we have so many different tumor models in the lab. We've got hot melanoma models, cold melanoma, immune excluded, ones that have too many myeloid-derived suppressor cells. We have, probably 20 different models where we all know they all have different barriers to immunotherapy. And so Rulan, who's a senior grad student in my lab, has developed this TLI avatar, she calls it, which is like an in vitro avatar of the tumor lymph immune interface. And so the concept is we establish all of this in the mouse. We don't try to build it from the bottom up. We just let it be however it is. And then we do the immunotherapy part ex vivo. And so we can give a vaccine or CAR T cells or cytokines or whatever we would want, and we can screen, for every mouse, we can screen like 50 different things. And so you have a really high throughput way to compare not only amongst one tumor, one animal, all these different therapies, but also between different types of tumors. And then the idea is to then go back and correlate. And so this is one of the things that we're very interested in doing. We're collaborating with Jeff Hubbell's lab on this because he's developed a whole library of matrix-binding immunotherapeutic molecules, and many of them, like I mentioned, here are ones that we've tested just here. They're all matrix-binding forms, and the idea is that in vivo they would target the tumor, but for each one of these you would want to test it in multiple different tumor models, and also in combination with checkpoint blockade. And so this is what we can do in our in vitro model. And so just to show you an example, in vivo here on the right, this is what their lab published. They developed CBD is not cannabis. It is an unfortunate name for collagen binding domain, which is the little, the engineering part that they did. So they made an IL-12 molecule that when injected, it will localize to the tumor, and this is their in vivo data. And so the only way IL-12 works is by activating the cells that are already there in the microenvironment. You would never be able to test this in like an organoid model because, it's not killing tumor cells, right? But the fact that we could recapitulate this in our TLI avatar, we were really excited about. And we've, as I mentioned we've now validated many of these with the tumor models that we do have in vivo data for. And so this is just an example. We can look at tumor cell killing over time. We can measure cytokines, things like IFN gamma, TNF, granzyme, and we can evaluate at the end by flow cytometry if we want, just like we can in a mouse. And we also asked, could we look at, could we recapitulate effects that we see only when we modulate in vivo the tumor microenvironment? And I'm going to talk about this next, but a big part of my lab is looking at lymphangiogenicis in tumors. And one of the things that we've grown to understand in the last 10 years is that non-lymphangiogenic tumors are less infiltrated. And the same exact tumor, when it's secreting a lymphatic growth factor, VEGF-C, the whole microenvironment changes to be more immune infiltrated, but also more immune suppressed. And we had published back in 2017 this surprising finding that even though lymphangiogenic tumors are more metastatic, they are very much more responsive to immunotherapy. And so this was some data from that paper where if we gave this as adoptive cell therapy, and so think of it like CAR T cells, kind of we just add activated T cells in its therapeutic setting, and we could see the lymphangiogenic tumors regress and stay regressed, whereas the non-lymphangiogenic ones grow back. We could also give a vaccine to the draining lymph node, which is, in this case, just a CpG in the protein that the tumor was expressing, and we see the lymphangiogenic tumors are completely cured almost. So we repeated these experiments ex vivo. So all the immunotherapy was done ex vivo. And you see like really very similar results. We see that the lymphangiogenic tumors in vivo respond much better to immunotherapy ex vivo. This is both for vaccinated and for adoptive cell therapy. And we could see, much stronger correlates. For example, here, like interferon. And we saw much more T-cell infiltration when we did a VEGF-C, an adoptive T-cell therapy. So basically, that was an example of sort of how engineering, in this case, tissue engineering, really, a lot of the engineering that went into this was million little details. How do we keep it perfused over long periods of time? How do we have a scaffold of support? What kind of matrix? Everything like that. But now we can actually use it to both. Well, if we can recapitulate responses in mice that have to do with the tumor microenvironment, then we hope that this will be very useful for patients as well. And we're starting now with head and neck cancer patients where we're getting samples of the tumor and the draining lymph node, and we're trying to do immunotherapy screening. But even with mice, it's amazing because we can start to understand what features of the tumor microenvironment correlate with response to these different therapies. All right, so next I want to just talk about immunophysiology because I consider physiology as sort of like bioengineering and biomedical engineering in a lot of sense evolved. One branch of bioengineering evolved from physiology, right? And so I think one branch of immunoengineering is evolving from immunophysiology. And this is what I really love the most. And I'm now going to talk about lymphatics. I've been studying lymphatics since I was a grad student, first in the biomechanical sense and the biotransport, but rapidly realized that their main function is really immune functions. And so that's where I've moved in the last 15 years. Lymphatics really, people think of them as just fluid drains, and if the lymphatics are blocked, you swell. But they actually are much more than that. They are actively regulated, meaning that lymphatic endothelial cells can sense anything in the microenvironment, cytokines, toll-like receptors, danger signals, toll-like receptor ligands, and they can modulate what they pick up and how they pump, what they're bringing to the lymph node. They can stop altogether if they want, and they can increase flow if they want. Anything really large that's in the tissue, and by large I mean pretty small, like a protein size or bigger, really depends on lymphatics to move through the tissue and to be systemically distributed. Why is that? Well, in this video, I'll just explain it before I play it, but this is just a live mouse here, and we put fluorescently labeled antibody that binds CD31. And it would bind blood vessels, who you can see here in black, and also lymphatics, which you can't see yet, and the thing is fluorescent, so that's why it's the background. We just pipetted it on an open ear skin, open the ear, and this is just, an antibody. But what's really striking is, so over the course of about 10 minutes, you can start to see the lymphatics very clearly. And you don't see the blood vessels. So if this mouse was dead, it would stain the blood vessels very strongly. Instead, it's being swept into the lymphatics. So it's like, the lymphatics are really pulling everything from the tissue, and that just shows that, at least for that antibody, the Peclet number is large enough where convection is dominating. And this really is so most factors that cells secrete, and in cancer we think a lot about things like exosomes as well, those are much larger. And so those all depend on lymphatics for transport. Okay, of course, everything that goes into a lymphatic ends up in the lymph node, and we're starting to think of lymphatics not only as just being needed to absorb the extra fluid that leaks out of the blood, but actually flipped on its head, which is maybe blood vessels leak to flush the lymphatics. And by doing so, you have constant surveillance of your immune microenvironment. So you have a mouse doesn't have that many lymph nodes, but we have almost 1,000. And every lymph node or lymph node cluster is specialized and knows everything about that tissue it drains. And it has all the information, all the antigens. There's so much going on there to maintain tolerance to your autoantigens. Every time you have an infection, you imagine, we get, like blood vessels become hyperpermeable, which flushes stuff into the lymphatics. But we can also, if it's an infection, we can get danger signals as well, and you have tissue breakdown products. And so, you have your own self-antigens and danger signals, so those are when you're most likely to be at risk of developing autoimmunity. And so this drainage is tightly regulated so that there can be different waves of presentation of antigens so that tolerance can be maintained between self-antigen, which is rapidly going in, and the pathogenic antigens, which are more picked up by these dendritic cells, and then go to the lymph node much slower, okay? And so, of course, once they're in the lymph node, then adaptive immunity can occur. So I'm going to focus on cancer now, because tumors solid tumors, lymphatics have been the most well studied in the context of solid tumors. And that's because it's been recognized for a very long time that a lot of cancers metastasize through lymphatics. And early in the field of lymphatic biology, once lymphatic growth factors and receptors were discovered, the first thing people looked at were cancers. And of course, it was found that the tumors that are more metastatic are the ones that are expressing more lymphatic growth factors, and they're engaging more with the lymphatics. And it is always considered that lymphatics are an escape route, so more lymphatics, easier to escape. But everything I just told you is like the whole function of the lymphatic drainage is for immune surveillance. And so why is that? When there's more engagement, you have more metastasis. There's actually bigger questions that have come out also in the last 10 years. We now know that lymphatics expand in all types of chronic inflammation, lung infections, asthma, inflammatory bowel disease. Even after vaccination, your lymph node lymphatics undergo massive expansion. And we don't really know why. When I say why, it's like, what is the function of that? There must be some function. What do expanded lymphatics do? And how does that help or hurt the immune response? And I really feel like this is a fundamental question, because if we want to control immune responses, we know how to we're learning how to do things like vaccinate or make the strongest immune response possible against cancer, but we're not so good at understanding how to fine-tune immune responses. And even our cancer immunotherapies are really, really extreme. Autoimmunity is a big side effect of a lot of those checkpoint inhibitors. So, unfortunately, this is in the middle of a bunch of fields. The lymphatic research community has done an amazing job at understanding just the molecular signaling pathways that govern lymphatic development and growth. But in terms of more on a systems physiology level, we still really don't understand it very well. And so, the questions that have been driving my lab has been really, how is lymphatic transport, activation, and expansion affecting immunity? Okay, and so I'm just trying to, give you a bird's eye view, but really, if we can understand why lymphangiogenicis correlates with tumor progression and metastasis, and how it affects the immune surveillance, and of course, how it alters the immune microenvironment, then I think we can make new discoveries in lymphatic immunobiology, which is really critical to understanding how to manipulate lymphatics for immunotherapy. Right now, lymphatics are not really a target for anything. There's no lymphatic targets, really. There's maybe some now new ones for the gut, and that's only for drug delivery to bypass the liver. But in terms of modulating lymphatics, which are really central to communication of immune information, we have no idea how to do it. And so this I feel like is a big opportunity. But of course, engineering, really anybody can work in this space. And so sometimes it seems like the work we do isn't really engineering anymore. But our goal is immunotherapy and manipulation, but we have to do a lot of basic work to get there. Okay, so I'm just going to summarize about 10 years of work for what we've learned about how tumor lymphangiogenicis, with a cartoon, of course. I was like, oh, do I put in data? And it was just too long, so I just decided to summarize everything. So we've looked at how lymphangiogenicis affects the tumor-draining lymph node. And in about a series of 5 different papers, we showed that, that first of all, lymphangiogenic tumors have far more antigen-specific T cells against tumor antigens in the draining lymph node than non-lymphangiogenic tumors. And this is because there's tumor-draining exosomes. The lymphatics are the main source of exosomes. And there's tumor antigen in the debris and everything that in cells that are picking up dead tumor cells, so macrophages and dendritic cells. So there's just a lot of antigen presentation in that draining lymph node. So it is very immune educated. And as a consequence, these lymphangiogenic tumors have more T cells that infiltrate because they're more recognized, okay? So that would seem like, okay, great, lymphangiogenic tumors should be more destroyed, but we know that they're more aggressive. What we also know is that there's a lot more transport. Exosomes, as I mentioned, we had a paper last year in JX Med where we looked at human exosomes from patients undergoing lymph node biopsies for melanoma, and they were incredibly enriched in this draining lymph, and they were full of melanoma-relevant exosomes that are shown to help drive a pre-metastatic niche. We also know that immune suppressive cells follow immune cytotoxic T cells. So you wouldn't have Tregs if you didn't have cytotoxic CD8s. And so this tumor survives if it can attract a lot of suppressive cell types and maintain a tolerogenic microenvironment. And that's exactly what the lymphangiogenic tumor does. Tumor-associated lymphatics, when they're activated, are incredibly immune suppressive. They're a big source of IL-10, IDO, TGF-beta. They also secrete INOS. They attract myeloid-derived suppressor cells. They attract Tregs. Also, because of the TGF-beta is also driving, as most of you know, fibroblast differentiation into myofibroblasts, which are really, myofibroblasts and cancer-associated fibroblasts are almost the same thing, and that leads to a stiffening of the tumor microenvironment, which also helps promote invasion. And so, you know, basically the lymphatics are playing on both teams. They're a yin and a yang. By pushing, you know, they're both driving the immune recognition. It's like a double spy. And then they're also promoting immune suppression so that these tumor cells can escape the immunity. All right, so as I mentioned, we did look at then how this microenvironment would affect response to immunotherapy. And what I didn't mention, I told you we had lymphangiogenic tumors and non-lymphangiogenic tumors, but we used a blocking antibody against VEGF receptor 3, which had been tried in clinical trials as a monotherapy and failed. It was designed to maybe help treat metastasis. Okay, so when we gave lymphangiogenic tumors so we made the tumors lymphangiogenic, then we blocked lymphangiogenesis or not. And that's where we saw these effects that I showed you. And this lymphangiogenic potentiation was really, really striking. We also demonstrated this not only with these types of immunotherapies, but with peptide vaccines, dendritic cell vaccines, checkpoint inhibition, and we used multiple models. So this was really a robust response. And what we now know is that one of the main reasons that lymphangiogenic tumors are more responsive to immunotherapy is because of antigen spreading. And that means that even if you vaccinate against one particular antigen, you activate so many other types of T cells with different antigens, different clones that also recognize tumor cells. And so, for example, in this study, we had an OVA-expressing tumor. We gave adoptively transferred T cells that recognize that. It's OT1 cells. They're basically T cell receptor transgenic mice where you take out the T cells and you activate them and they all recognize OVA. And so they go and kill the tumor cells. They only recognize OVA. So if we then analyzed the tumor afterwards and looked for all the effector CD8 T cells that were activated and did TCR sequencing, we saw far more clones that were these are all non-OVA clones, far more expanded in the control groups. These are the lymphangiogenic tumors, as opposed to the ones where we blocked lymphangiogenesis. And this was really a striking difference here. If we look at humans, serum VEGF-C before immunotherapy, which, remember, serum VEGF-C, lymphangiogenicis, is always bad in the absence of immunotherapy, but actually it was very good in patients. So the patients that did have high VEGF-C levels in their serum had a better response. This was just a peptide vaccine, and we were looking at CD8 T cells that expressed the TCR for melon A, which was what the vaccine was. And you can see they were much more responsive, but even more interestingly, in a checkpoint blockade trial that was done by BMS, we had access to the blood before they started the trial, and those patients with high levels of VEGF-C in red had 90% survival after a year, whereas everybody else was about 50%. So, I mean, it really is not just a mouse effect. So this was really exciting. And so these are basic discoveries, right? And it took us, and I just wanted to point out here that if you look at survival rates for non-immunotherapy patients, it's the polar opposite. So it's not like lymphangiogenicis is better for survival anyway. It's not. It's the opposite. And so this was, we finally got to this place after trying to understand for so long what lymphatics are doing to the tumor microenvironment, how they're changing the immune infiltrate, what exactly they do to, are they driving T cell exhaustion, looking at all of these things, and then finally getting to this point. And since we published this paper, we have done a lot of work in trying to understand the mechanism. We had a hypothesis, but I just want to show you again in a cartoon what we're finding the mechanism is. And there is, again, I wanted to give a general talk, so I didn't put very much data on here, but some of this is published and some of it is in revision. Basically, as I mentioned, a lymphangiogenic tumor is secreting VEGF-C. It's immune suppressive. One of the things we found was that CCL21 is strongly upregulated, and this is a cytokine that is secreted in the lymph node. It's called lymphoid, it's SLC, secondary lymphoid chemokine. That's its other name. And it is critical for bringing in the immune cells before education occurs. So if you have an infection or you have a swollen lymph node, CCL21 is just super upregulated. The idea is you have dendritic cells there, and they need to find their cognate T cell partner, and so you want to attract as many naive T cells as possible to go in and out and sample as many to find that right T cell. And so CCL21 is really critical for T cell education. And the cells that have the receptor for that are cross-presenting dendritic cells and naive T cells. Of course, there's some other cells too, but those I'm going to focus on. So here you have like perfect environment for T cell education. We also see some stromal changes, like lymphoid-like stromal transformation, we call it, and this includes upregulation of high endothelial venule markers on the surrounding blood vessels so that the T cells actually can exit and enter the tumor, and also proteins that are necessary for the rapid communication between these two cell types. And so this is we know that lymphangiogenic tumors look like this before we do immunotherapy, okay? And once we induce T cell killing or any tumor killing, we get a cytokine shift from a suppressive to a cytotoxic. We also have an immunogenic release of tumor antigens, which are picked up by the cross-presenting DCs, which can then start to mature. We also have an upregulation of CXCL9, and this is driven by IFN gamma. This is by lymphatics again, but only with IFN gamma. So without the T cell killing, you wouldn't have this upregulation. And so we're also bringing in now more of the activated T cells. And because of all of this, we can get local DC activation, picking up the tumor antigen from all of these dying tumor cells, and those can start to activate all those naive T cells that are going in and out. So we get all this bystander activation, and this is what drives the antigen spreading. And we did a lot of experiments to really tease out these mechanisms. For example, blocking egress of T cells from the lymph node, blocking CCR7 before immunotherapy, and also in vitro, in the very first, the in vitro model I showed you, we're actually using that now to further tease out these mechanisms because we have so much control in vitro where we can manipulate one cell at a time and compare everything. And so, a couple of things. Well, first, okay, that's really interesting. How do we actually use this now for therapy? We finally found something super interesting with lymphatics that could be potentially translatable. So the first question is, does lymphangiogenics potentiate other cancer therapies that induce immunogenic cell death? Turns out that radiotherapy induces immunogenic cell death. And this has been well studied in the last 10 years. And you can actually get some immune response just from radiotherapy because these tumor cells, when they're irradiated, they die. They die in an immunogenic way. They're releasing their tumor antigen, and immune cells can come and pick it up and activate other cells. And so Maria, Stella, and Nick, who are both postdocs in the lab, but not anymore. They're both working in industry now. They were looking at this and found that lymphangiogenic tumors are indeed more responsive to radiotherapy by a lot. These melanomas, these are just B16, they're not expressing OVA. These melanomas are very resistant to any kind of therapy, including immunotherapy. But if they were expressing VEGF-C, they were super responsive to radiotherapy. And we could do this either with VEGF-C overexpressing or by blocking this VEGF-C receptor. So that's one thing. We found that this was correlated, as expected, with an immune response, that the VEGF-C tumors after radiotherapy had a lot more of IF and gamma positive T cells. You can see here, this is a quantification. They were more activated, and you can see just like, just if you look at these differences in the percents that are activated, it's really huge. We were really surprised. And even like lymphangiogenic tumors could potentiate abscopal effects of radiotherapy, which is, again, really difficult to see in mouse tumor models. And for those of you that aren't familiar with that term, it just means that you have another, if you have multiple tumors and you irradiate one, you can activate an immune response that will kill another tumor that wasn't irradiated. And so we could see that here. You can see here's the primary tumors, looks similar to what I showed you, but then here's a contralateral tumor that was not irradiated. No, this was just in, and those tumors were also completely, well, almost, had very, very good response. Okay, so now last is how do we translate this? So now that we know radiotherapy can mimic these effects, and we know that if this mechanism that I showed you is really what's going on, so Maria Stella worked on this a lot, and she tried a lot of different things. One thing we tried was to induce lymphangiogenesis within the tumor site to promote immune infiltration. The problem is that that's very closely associated with metastasis as well, and we thought, we did it, we showed that it worked, but we were worried about further translation just because it was too close to like having bad effects of metastasis. So that was the downside. And then since this abscopal effect worked so well, that just indicates that we were really activating an immune response in that local tumor environment. So could we do that in a tumor vaccine? So the idea is just what I showed you with the abscopal effect, but instead of irradiating the tumor, we take the tumor out, irradiate it, transfect it to express VEGF-C, and then implant it. So there's no chance that could metastasize itself. And so they would be in two separate places. The lymphangiogenic vaccine could be anywhere. And this actually kind of approach has been well developed, actually. The approach of a whole cell vaccine, I mean, this is basically this is how standard vaccination works. You take a pathogen, you attenuate it or kill it, and then you inject it with adjuvant, and that's a vaccine. We're doing the exact same thing, but with tumor cells. And the thing that we're doing that's unique here is that these tumor cells are expressing VEGF-C as they're dying, and so they can engage with the lymphatic system and take advantage of that, and hopefully we could see antigen spreading. And so this has been developed with GM-CSF. By Glenn Drainoff. It was in phase 2 clinical trials at Novartis. It's just that GM-CSF is kind of too general, I think. I think the efficacy just wasn't there. It was safe, though. Okay, so this is the concept. We hope that these dying tumor cells would have the same effect as what we saw in our tumors and lead to this. So we did that, and first of all, you can see, indeed, these tumors, even though they were dying, within a week, they could induce lymphangiogenosis. That's shown in red here. And we could see all kinds of green infiltrating T cells here. I'm sorry, I forgot the legend here, but green are T cells. So we had lymphangiogenosis in that site. We attracted T cells in that site. And interestingly, as before, those T cells were really enriched only in the naive component. Okay, and that means that at least we had the right setup for what we were hoping to accomplish. If we then added, we let these vaccine sites, we let the injected irradiated cells sit there for a few days, and then we added adjuvant. And in this case, we just added imiquimod and a CD40 agonizing antibody. And you can see that even if we just look at this antigen spreading concept, you can see really a lot. We only looked at 4 possible antigens because that's what we had tetramers for. TRP2, GP100, those are common melanoma antigens, but then these 2 are mutated neoantigens that have been shown in this B16 model. And you can see that just in these 9 or 10 mice on each, there were every mouse in the VEGF-C VAX group, almost every mouse had multiple antigens that they had T cell reactivity for. So what you do is you take out all the T cells and then you just re-stimulate them with, this is an ELISpot assay, you re-stimulate them with the peptide, and if they react, then that means that they were specific for that peptide. So even out of these 4 we looked at, there were far greater numbers of different diversity of clones among these 4. And remember, the only difference between these 2 vaccines is just the presence of VEGF-C. Everything else was there, the adjuvant, the tumor cell lysate, everything. And if we did an experiment where we looked at protection against another implanted tumor, and we used GM-CSF as a control, because that's the one that's been in clinical trials, that's called GVAX. We can see huge differences with the VEGF-C vax, both in terms of the T cells that get activated, and actually none of these tumors, 9 out of 9, I should say, all of the tumors, none of the tumors ever grew. So it was 100% prevention. And GVAX was very similar to our control vax. And then the pandemic hit and we couldn't access the mice for a long time, but after a year almost, you can see this is day 320, Maria Stella went back in the lab and she's like, what, these mice are still here. We were doing a survival study. They're still alive, all of them. What do we do? And so she decided to re-challenge them and then see. So this is a year later, and again the VEGF-C vax showed at least, the ones that survived the first challenge, half of them survived the second. So they had good long-term memory. And then therapeutically, we could also see very strong effects, and especially when combined with PD-1 blockade. So basically, this is what we're now moving towards clinical trials. We're starting with veterinary animals because there's a lot of dogs that have cancer, and they're patients just like people, and so this is a whole cell vaccine that that could be used for any kind of tumor, right? I mean, it's not like we have to make a molecule that's specific for a dog protein or something. So because it takes the animal's tumor, the person's tumor, and then turns it into a vaccine. And so this is where we're about to embark on with collaborations at the University of Illinois. And so to summarize, tumor-associated lymphatics, they're very interesting. They regulate the inflammatory microenvironment, but they are they make the tumor very primed to be responsive to immunotherapy or any type of cytotoxic effects. And so understanding this balance, could lead to completely new strategies for immunotherapy. I think understanding this in the context of other types of chronic inflammation, including most autoimmune disease, will also help us be able to fine-tune immune responses better, and that's a whole other area that would be ripe for people to go into. Okay, and so again, I just wanted to, just say I hope this vignette kind of exemplified the road from basic science discovery to immunoengineering technologies, or at least invention or, development. But it doesn't have to be one way. You can both discover and develop and then discover again and use the tools that you develop to make new discoveries and on and on. So with that, I'd like to thank you for your attention. This is my lab. I mentioned people who did the work as I went. These are my collaborators. Ralph is a radiation oncologist. He's been amazing. Funding sources, and answer any questions that you would like to ask.
Please wait for the microphone to come around, and make sure you speak into the microphone with your question.
Thank you for a great talk. Is the plasticity of lymphatics solely dependent upon their environment, or are there different precursors that kind of are hardwired to function in a particular way in a particular environment?
That's a great question. We don't know, we used to think that we didn't really see that much turnover of lymphatics until we looked at areas of chronic inflammation. The plasticity under normal development, once I've only studied these in mice. Once the mouse is an adult, we do see lymphangiogenicis occurring in the ovaries, for example, during cycles. And then they also turn over a lot in the gut, it turns out. So lacteals absorb all of the lipids in your gut, and they're in the cilia, and those are constantly turning over. In terms of other plasticity, there's debate. So in the lung, if you induce lymphangiogenicis and then you resolve the inflammation, sometimes the lymphatics just stay around. The expanded lymphatics stay around for a very long time. But there's debate in the literature, so sure.
Nice talk. I have two questions, one basic and one translational.
Okay.
So in your model there, you're showing most of the antigen presentation occurring in the tumor itself, whereas we're taught that that's actually occurring in the tumor-draining lymph nodes. And I'm wondering if you have evidence that's showing that that's all occurring in the tumor and not in the draining lymph nodes.
It definitely happens in both.
And then the second question, so we have a tumor-infiltrating lymphocyte therapy going on where we're doing some CRISPR editing. Would it be reasonable to throw in a VEGF-C gene into the TILs and then reinfuse them? Or would that actually be worse for the tumor?
That's hard to say, because I think that what lymphangiogenicis does, and we're still trying to understand whether expanded lymphatics also benefit post-immunotherapy, and they could, for example, they could help support memory T cells or keep the the T cell plasticity better so that they don't get terminally exhausted so quickly. But most of what we've looked into, the benefit is prior to immunotherapy. So the idea is it primes the environment to enhance. Yeah, so if T cells that were coming in and I don't know if that would, I don't know. It's an interesting thought, but we did experiments in that paper, that Science Advances paper, to show that most of it, a lot of the differences between lymphangiogenic tumors and none was actual in situ education using things like FTY720, which blocks egress from the lymph node, but there was still certainly T cell education in the draining lymph node as well.
We have a question from the online audience. What is the impact of lymphoangiogenesis on the metabolic fitness and stress of immune cells in tumors?
We don't know. It's a great question. Yeah. In the lymph node, I will mention, lymphatics are the main source of IL-7, which is, keeps, it's a survival factor for memory and naive T cells. And so, the question is, are they also doing that in the tumor microenvironment? We don't know.
Yeah, just to continue on the epitope spreading question, I'm just wondering mechanistically, do you think it's really related mostly to more naive T cells getting in, or is there a link where the killing that you started is releasing more antigen, and it's mostly this new antigen population that's available? Or do you need both?
Well, that's why we used a lot in the early studies ovalbumin expressing tumor cells exactly for this reason, because then we could do adaptive T cell therapy with ova. So that killing will be the same in both tumors. So the difference was that these other tumors had more naive T cells just at steady state. One of the things we did to that end was we blocked CCR7 just before immunotherapy, and that would block the infiltration, and we showed that blocked the infiltration of the naive T cells, and we completely lost the immunogenic potentiate. Then the lymphangiogenic tumors responded identically to the control tumors. So it really did depend on that signaling in the beginning. So I think it's the advantage, obviously all this stuff is going on, but the added benefit of lymphangiogenicis is probably, at least one main reason is this.
Hi, thank you for that great summary, Melody. I have two questions. The first is, when you're inducing lymphangiogenosis, what happens to angiogenesis?
It's a very good question. We always look at blood endothelial cells, and we sometimes see a little bit more, but not that much. One of my slides had that. I don't want to I can show you after. But we always look at that, and we don't see big differences, really. The thing is that VEGF-C, we use VEGF-C, and there's also, we could use VEGF-C 156S, which VEGF-C has some affinity for VEGF receptor 2, but VEGF 163S is mostly targeting only VEGF receptor 3, and so we could probably address that more specifically. In the human populations, though, with the response to immunotherapy, and when we looked at VEGF-C in the blood, we also looked at VEGF-D and VEGF-A, and VEGF-A was not at all correlated. Okay. So,
all right,
yeah.
Thank you. And my second question is, how do the lymphatics behave in the hypoxic core of the tumor?
We don't see lymphatics in hypoxic core. Usually the lymphatics are in the margins, and it depends on the type of tumor. If it was an acinar structure like breast cancer, we generally just see them expanded around the margins. It's not like they're growing in, because they don't need to. It's not like the tumor needs blood vessels to have oxygen at a certain density. The lymphatics just need to be collecting around it.
Okay, thank you.
Yeah. In melanoma, we see more density everywhere, but it starts out very, skin is really different. Okay.
Yeah, thanks, Melody. I was just wondering about the VEGF-C then. It's kind of seemed like a master regulator. What makes it go on or not in a given tumor?
Well, this is it's interesting because these questions are all amazingly great questions, but there's so little known about these things. So we did have to look at, what is driving it, what else is going on, and why. Turns out when we looked at chronic inflammation, other types of chronic inflammation, and we see the same thing happening, it's always sort of part of the Th2 response. M2 macrophages secrete a lot of this. It's part of the kind of almost a resolution response. And so, it's really myeloid-derived suppressor cells and macrophages that are the main source. But other cell types have been noted to be able to secrete them.
We have
last question, maybe from a student or a trainee. Anybody out there that wants to ask a question?
Great talk. I'm wondering if we know how TLR agonists that are frequently included in these cancer vaccines might modulate lymphatic drainage or the lymphatic endothelial cells, or with the advent of mRNA vaccines, if you've thought about how those might modulate drainage.
Wait, sorry, the first thing you said was what kind of agonist?
TLR agonists,
Oh.
like CPG or,
Yeah, I mean, there's so many questions there that we really don't know exactly what, we're looking at adjuvant effects right now and whether you use CPG, which is TLR9, intracellular versus LPS, and we don't know, I'm so sorry, but it's a good question. The effects might just be too subtle to be obvious.
I have one quick question,
Okay.
and then we'll wrap up. I'm wondering, on this comment about autoimmunity, which is the flip side of all of this, is there a thought about going after immune tolerance induction, which is a huge issue with, like, organ transplant and things like that? Is that a part of what you're going to learn, do you think?
Yeah, so it's very interesting because also transplant rejection can also be associated with lymphangiogenesis. But it actually makes sense because it's sort of like what we have when we have immunotherapy. You just need initial cytotoxic environment, and then the lymphatics are actually helping the immune system destroy it. What we have done, we have actually done some work there, and one of the things we did was if we first block lymphatic transport, by photodynamic therapy, it's like where you put in a phototoxic drug, let it get taken up by the lymphatics, and then just shine a light at a certain frequency, and then it destroys the lymphatics, and they get all clotted up. Then it's an immune privilege site, and then we did transplants of like heart, you can take an embryonic heart, cut it into pieces and stick it in an ear, strangely enough, and it will beat, and you can watch it beating, and then it gets rejected when it stops beating. So it's kind of an amazing model, and we could do that if we block lymphangiogenesis, but if we block lymphatics, period. But then it only lasts as long as the lymphatics are blocked. So we've tried this other strategy where we block the lymphatics, but then locally try to expand so that there's no drainage, but the immune suppressive effects are there. And then it probably has, it's tricky, but I think it has to probably do with the timing and kinetics of antigen delivery, because you want it to be, does that make sense? Like,
Yes.
it's almost like when people give peanut allergy treatments and they give tiny microdoses. It could have to be with how much and when or how little, antigen is delivered to the lymph node. Okay. But we don't really know.
Well, great.
They're good questions.
Well, let's thank our speaker. And Dave and I are going to present Melody with a plaque.
Thank you.
And maybe we could get a picture here. You guys can have